Hi, my name is Nicole, and today I'm going to tell you a story about identity theft. Centromere identity theft. This is a very serious crime that can lead to tumor development, developmental abnormalities, and even miscarriage. So, who are these imposters lurking around causing mischief? Well, they're known as neocentromeres. Normal centromeres are the good guys in the story because they play a very important role during cell division. They help to make sure that the chromosomes get properly divided between each new cell. Neocentromeres, however, are very sneaky. They look and act just like the normal centromeres. So during cell division, these structures called kinetochores get recruited to both the normal centromeres and the neocentromeres. Next, microtubules attach to these complexes and they begin to pull the chromosomes apart. Eventually, however, these pulls from opposing forces causes the chromosome arms to snap off, and as a result, the neocentromeres have stolen precious genetic material from the chromosomes. Using Drosophila, the fruit fly, as a model organism, I want to assess the impact that these detrimental structures have in a germline of cells. But in order to do this, I first needed to create a model. My lab has recently made a breakthrough discovery. They've discovered a protein in Drosophila that is capable of essentially building a centromere. And I'm going to refer to this protein as protein C. So when protein C is present in a cell, it's able to gather all of the materials it needs, and it can basically build a centromere. So in this way, it acts as a construction worker. Using this piece of information and molecular cloning techniques, I constructed my own circular piece of DNA that codes for protein C. So if you think about it, that piece of DNA kind of acts as an instruction manual. I took that instruction manual and I inserted it into healthy, normal somatic cells so that these cells would now have all the information they needed to make a bunch of the protein C construction workers. I also genetically manipulated these cells so that those construction workers would only work on the very specific sites of the chromosomes so that they could build neocentromeres. Using fluorescence microscopy, kind of like using a UV light at a crime scene, I was able to see that those once normal, healthy cells had gone to the dark side. They had assembled neocentromeres, and this was incredible because my experiment worked. And it's so important that this works because we now have a technique that we can use to basically take this plasmid and make new cell and transgenic fly lines that form neocentromeres on demand. And this is significant because now we have a way to further study and assess the impact that these structures have in cell lines and in fly lines, because we really want to get down to the bottom of this case, because identity theft is no joke, and neither are neocentromeres.